Okay, so I would now like to officially welcome you to this third session of the BioClock Academy. First, my name is Hannah Brooks. I'm a BioClock PhD candidate, and together with Laura Pape, also a BioClock PhD candidate, we will host this session. For the people that joined for the first time, maybe just a small introduction. So the BioClock Academy series is organized by BioClock Academy, BioClock Consortium. This is a Dutch project, and the aim is to investigate the biological clock on 3 different levels, so society, nature, and health. And with this BioClock Academy, we want to address everyone who is interested in this field, but especially the young career researchers, because we want to provide them with a good knowledge basis and proper theoretical knowledge from experts in the field. So this lecture series is organized every 3rd Wednesday of the month. And for the people that were already here in the last 2 sessions, this time we will organize things a little bit different. So there will be no Q&A session afterwards for the BioClock trainees. This gives us a little bit more time for questions. So the basic idea is that we have a talk of 40 minutes, after which we can have a discussion from 15 to 30 minutes.
Yes. Okay. So also, hello from me. Nice to see that you're all here and to hear more about the topic today, which is about molecular clocks, zooming in on circadian rhythms. And this talk is given by Dr. Inês Chaves, who I would like to welcome very much. She is a research associate at the Department of Molecular Genetics of the Erasmus University Medical Center, and her research is centered around the circadian clock and the health risks that are associated with circadian disturbance. And she investigates adverse effects of circadian disturbance and adverse light exposure during development at the neonatal intensive care unit. And she's aiming to understand the underlying epigenetic mechanisms and identify associated biomarkers. So welcome and great that you're here. And you're very welcome to share your screen and we can start.
Okay, so I hope the screen is visible to everybody. So first of all, I would like to thank the Academy for the invitation. It's an honor to be able to present maybe the basics of the molecular oscillator and how things have evolved. So what kind of genes and proteins are actually responsible for our 24-hour rhythmicity. So today I would like to tell you about molecular clocks, and not only the molecular oscillator, but also to tell you something about genes that are involved in input to the molecular clock and genes that are responsible for the output. I would like to bring it also a bit in a seasonal perspective and try to show how the molecular clock is also responding to seasons. Very briefly, how this clock is also changing with age. And maybe if we have time, I would like to give you some insights into different functionalities of clock proteins and proteins which do not belong to the clock and how they have some kind of interchangeable functions. So I think this is something that probably everybody listening now knows, is that we are living on Earth, and because of that, we are imposed a light-dark cycle of 24 hours, which has led to the evolution of a circadian rhythm in most organisms. And this circadian rhythm is expressed in behaviors and in physiology, but of course, at the core, if we really zoom in, there's a bunch of molecules of genes and proteins which are responsible for driving this oscillator. I think you may remember from the other 2 presentations that we have our circadian system, And in order to have a functioning circadian system, we need to have an input to the system, which relies cues, external cues, which will allow synchronization of the system with the environment. But of course, these cues need to be acting on something, and that is our circadian pacemaker, the oscillator, which is the molecular oscillator. And of course, in order to have a functional clock, which is translated into physiology and behavior, you need to have outputs, which are also regulated by this circadian oscillator. So today I would like to focus on genes and proteins which are involved in each of these parts of the system. The largest focus will be on the oscillator itself, but now I would like actually to start by briefly mentioning actually the photoreceptor genes, which are present in different species and which are responsible for receiving the light input from the environment and transmitting it to our circadian oscillator. So we have here two pictures of a human eye and a Drosophila, a fly eye. And in essence, these are the same, but also very different. We have a eye, as we know, and the Drosophila has what's called a compound eye with several smaller ocelli. But in essence, what's important in these eyes is that there are photoreceptors present in the retina. And there are different types of photoreceptors in our eye. There we have the rods and cones, which are responsible mostly for the visible perception of light. And we also have at the retinal ganglion cells, the melanopsin photopigment, which is mostly responsible for the circadian photoreception. Joke Meyer already went into depth about this, so I just here briefly wanted to mention what are the type of proteins, genes involved. So we have in different species several photoreceptors, which could be responsible for the circadian input into the system, but they could also be just responsible for inputs such as phototropism in plants, which means that the plant will, for example, turn to the sun when there is light. So we have, for example, in cyanobacteria, we have phytochromes. In insects, we have rhodopsin and cryptochromes. In vertebrates, you have opsins and the melanopsin I already mentioned. And in plants, for example, you have also phytochromes and cryptochromes. What is interesting here, and that will become evident later on, is also that, for example, the cryptochromes, and you may know or have heard about that already, they're also actually clock proteins. So these seem to be proteins that actually play a role in photoreception, but in some organisms, they also really play a role in the core oscillator. And these intercrossed or correlated functions are part of the evolution of the circadian clock. So we know that we have a handful of genes which is responsible for the input into the circadian system. But what is driving actually our circadian clock? What is at the core? What is at the core of the molecular oscillator? And so if you think very basically of the molecular clock, of the molecular oscillator, the basic principle is that you need to have a feedback loop where you have activator and repressor molecules, which in turn activate and repress each other, creating a feedback loop. So this is a very schematic view of this, where you have two proteins that activate each other. And this feedback loop is also connected to an output and is connected to the input, either via light, for example, as is shown here, or via temperature changes. And what's important is that this oscillator can respond to cues, but also can generate an output. And in evolution, there are very different organisms which also have very different molecular oscillators. This is quite a complicated view, I think, at the moment. So I'll try to guide you through, but basically what I want to highlight here is that all species, from cyanobacteria to mammals, they have evolved a molecular oscillator, which is always based on feedback loops, but the type of feedback loops is different. For example, we have here on the left, top left, the cyanobacteria circadian oscillator, which is based just on 3 proteins, the Chi A, B, and C. And basically, it's a phosphorylation feedback loop where Chi C is phosphorylated or unphosphorylated, and depending on the status, you will have different outputs, and this will be occurring rhythmically. Now, if we move on from cyanobacteria to fungi, for example, Neurospora crassa, which is also a model which has very well been studied at the molecular level, we see that we now have a clock which is also composed of feedback loops, where we have the white collar complex composed of white collar 1 and 2 proteins, which are also themselves photoreceptors, which is then activated by, or is activating the freak gene frequency. And this freak gene is actually creating a feedback loop on itself. And important here is that we have a combination of a transcriptional feedback loop and also a clock, which is linked to a feedback loop in terms of localization in the cell. So the frequency complex or the white collar complex will shuttle between nucleus and cytoplasm, and that will be also part of the feedback. And of course, this transcriptional translation feedback loop theme comes back in most of the organisms further in evolution. And in plants and animals like insects or vertebrates, you also have the transcriptional translation feedback loop system composed of slightly different genes, but being in the base is the same. So in Drosophila, in Arabidopsis, for example, you will have the CCA gene, which is very important. And in Drosophila, in animals, you start having like clock and cycle or clock and bimal in mammals, which are the positive activators, which then activate transcription of the repressors and so on. So basically, all molecular oscillators are composed of feedback loops, and these feedback loops have evolved from just protein-based post-translational modification loops into actually more complex transcriptional feedback loops combined with either nuclear localization or post-translational modification loops. I can't move. Why? And for example, one example is if we compare the human or mouse circadian oscillator with the one of the zebrafish, is that in the core they are basically the same, but in the zebrafish we have like a duplication and much more than that of some of the molecules. So instead of having 1 clock or 1 bimal molecule, you have 3 or 4. So in evolution, a large redundancy within the circadian core oscillator proteins has been created. And that also shows that the circadian oscillator is very important. So this is just a highlight of how the circadian oscillator functions. And now I would like to go more into detail how the mammalian oscillator works and what are all the proteins involved and how this transcription translation feedback loop works. Okay, so I start here with the cry genes, the cryptochrome genes. So you heard me earlier on mention that cryptochrome is in plants and Drosophila a light-sensitive protein. So what we have is that in mammals, the cryptochromes do not have any photoreceptor or light-sensitive function, but they are really core clock proteins. And once they are transcribed and translated into the cytoplasm, that occurs because there is an activator complex, clock and bimal. These are the 2 activators of the mammalian oscillator. Clock and bimal are 2 transcription factors, pass BHLH transcription factors, which form a heterodimer and bind to E-box elements in promoters of clock genes, such as the Cry1 and Cry2 genes. Upon binding, they will activate transcription of the Cry genes, which then, together with other clock genes like the period 1 and 2, so Cry and Per will be transcribed, translated, and they will shuttle back into the nucleus, where they will form a heterodimer. And this heterodimer will then repress the activity of clock and BMAL. So they repress their own transcription by inhibiting the action of the activators. So we don't only have these clock genes. There's also reverb alpha, for example, which is also a transcriptional repressor, which is also containing an E-box element in the promoter and is also activated by the clock BMAL heterodimer. And this reverb alpha will actually feedback on the activators and is repressing transcription of clock and BMAL. So now here we create a second feedback loop. And although the whole circadian oscillator is formed by negative feedback loops, within the mammalian clock, we always talk about the negative loop, which is the one that regulates the negative regulators of the clock. And we have the positive loop, which is actually regulating the activity of the positive activators. This is sometimes confusing, so I hope I'm making this clear. Now, I mentioned we have a circadian output, and if we only would have these about 6 clock genes, which should be rhythmically expressed, I don't think we would be able to have a very strong oscillating output. But what happens is that we also have a lot of what's called circadian clock control genes, CCGs, and these genes also containing an E-box element in their promoters, which is also positively regulated by clock and BMAL and negatively regulated by the Kuiper complex. And this oscillating transcription of clock control genes leads to the oscillating output in physiology and behavior that we are familiar with. One thing that you may have noticed is some of these proteins depicted here have little flags hanging. For example, Cry and Purr here have little black dots, which represents phosphorylation. Phosphorylation is a post-translational modification, which is very important because it can regulate interaction with other proteins, but in this case, it's very important to regulate stability of a protein. We also see here acetylation, and for example, BMAL1 is being acetylated, and this occurs by clock itself, which is an acetyltransferase, and the presence of an acetyl group on BMAL is important for BMAL to be functional. So post-translational modification is very important for a proper functioning molecular oscillator and is also extremely important for the exact period lengths of a cycle. So we talk about the circadian clock, which has a cycle of approximately 24 hours and which is daily reset by light in order to keep really in pace with the exact 24-hour day of the Earth's rotation. And alterations in these post-translational modifications, which could lead to changes in proton stability, will actually affect the length of the circadian cycle. So I show here an example. So we see here a couple of circadian patterns of the Purr and the BMAL gene and protein. So the dashed lines correspond to the gene and the full lines to the protein. And we see that for Purr, there is a big difference, a few hours between the peak of RNA and the peak of protein. So RNA is formed and it reaches a peak, and the peak of protein will only appear a few hours later. And this distance is very important because if you change the stability of the protein, for example, if it will be more stable, the protein peak will appear earlier, and this will mean that your oscillator will tick faster. And the opposite would be true as well. So basically, period length is determined by timing of nuclear accumulation is one, but also by post-translational modification of clock proteins such as Cry and Purr. And of course, gene dosage, if you knock out a gene, you also will have effects. To demonstrate how these post-translational modifications can affect the period lengths, I'm showing an example of one of the first mutations that was identified in humans that was linking to the advanced sleep phase syndrome. This is a syndrome where patients actually have very early sleep patterns. So they fall asleep around 5 or 6 in the afternoon, and they end up waking up middle of the night. So their clock is actually having a very short period. And if you see here, ectogram depicting the activity or the sleep-wake cycle of these patients, you see that their period is actually shorter than 24 hours. So this is depicted in a 24-hour scale, 2 times. And if the period is shorter, you see that every day the person will wake up earlier. The mutation that was identified in this patient or in this family was a mutation in PER2, which was affecting the site that the casein kinase, which is a protein that adds the phospho group to the PER gene, is binding. And because of this, PER2 could no longer be phosphorylated at this position. And what this means is that PER2 was more stable, and the peak of protein would appear earlier than in normal subjects. And this would induce a shorter period, which is observed in the sleep-wake cycle behavior. Another example of how period lengths can be dictated by gene dosage is, I'm using now the CRY genes as an example. So when the CRY genes were identified as being part of the mammalian circadian clock, this was done by generating mice that were knocked out for either of the genes. So if you have a normal mouse, and we see here again one of those ectograms that you've already seen in previous BioClock Academy seminars, is that a normal mouse has a period of approximately 23.7 hours, very close to 24. And if we knock out the CRY1 gene, the gene for cryptochrome 1, we see that now we have a very short clock, which is just about 21.5 hours. And on the other hand, if we knock out the other CRY gene, CRY2, what we see is that we have an opposite effect on behavior, and we have a clock that ticks with a period of approximately 24.5 hours. So we see here that there is redundancy in the cryptochrome genes. We have CRY1 and 2. But if we knock each of them out, we actually have an opposite effect on the cycle length of the circadian oscillator. What's also interesting is that these CRY genes are actually essential sorry, for the correct functioning of the circadian oscillator, because if you knock both out, what happens is that if animals are kept in constant dark conditions, they no longer have rhythmicity. So the circadian clock is lost, basically. So we see that by missing CRY1, we actually have a very fast ticking clock. But what's interesting is that if instead of missing a gene, you have a mutated gene, for example, I'll show you now, you could actually find the opposite effect. So what we see here is an activity ectogram from patients from a family with delayed sleep phase syndrome. So earlier on, I talked about the advanced sleep phase syndrome, where a mutation in PER2 ensures that PER2 is more stable and the period is shorter. Here, what we see is that these patients have a disturbed sleep-wake cycle, and this cycle has a longer period. And what's characteristic of this syndrome is that patients, subjects, can only fall asleep very late, sometimes at 2 or 3 in the morning. And that means that they would have to sleep until midday at least in order to have enough sleep. And because our societal demands don't allow for that, these people usually suffer from depression. They cannot work properly. So it has a very high burden. And in this case, this was the first mutation ever identified for this type of disease. And it was a mutation in the CRY1 gene. And I mentioned earlier that a knockout of this gene would lead to a shorter period, but these patients have a longer period. And what happens here is that there is a mutation in one of the intron-exon splice sites, which actually leads to a protein missing one of the exons. And because of that, this protein is more active. It's a gain of function mutant. And that means that this would be the same as having more cry1. So less cry1 leads to a shorter period, more cry1 leads to a longer period. So I hope that it becomes clear that we do have the oscillator and that the exact protein composition is very important for the 24-hour rhythmicity. And that a critical part is that these proteins are modified post-translationally. They are phosphorylated, acetylated, ubiquitinated, and that all these transformations are very important for the stability of the protein. And that ensures that our circadian period is very stable. So this is what is the basics of our circadian oscillator. And of course, I already mentioned that we wouldn't have a real circadian physiology if this would be it. So now I would like to briefly go into what are other genes and how many of the other genes are actually oscillating. One of the first studies that has analyzed this, and we are now going back more than 20 years again, was a microarray study. So nowadays, if you want to analyze transcriptome, what you do is you basically sequence all messenger RNAs in a cell. But earlier on, that was not possible yet. And what would you have would be what we call microarrays, which would have small probes in each. So each of these dots would contain probes for one of the genes you were interested in. And by hybridizing RNA from a tissue, you would be able to see what the expression levels were. And by doing isolation of RNA at different time points throughout the day, you could look at different expression of genes throughout the circadian cycle. So what Panda has done was this microarray study both on SCN tissue and liver tissue from mice. And what he found out at the time was very surprising, is that more than 10% of our transcriptome is actually under circadian control. So the most recent studies show that even at least 50 to 80% of all genes are oscillating somewhere at some time in our organism. But at the time when the first results came out showing that 10% of the transcriptome was oscillating, that was quite striking. And what was observed, we see here a depiction in blue of oscillating genes in the SCN and in red of oscillating genes in the liver. And each graph contains the time on top. So ZT for Zeitgeber time, the time of the light synchronization. And we see that there are genes that will have a peak at a certain time of the day, beginning of the morning, for example. You have genes that peak middle of the day, and you have genes, for example, that will peak middle of the night. And the same is occurring in the liver and the SCN. So you have a lot of genes which are oscillating, but there is also a very large tissue specificity here, because there's only about something like 20 or 30 common genes that oscillate in all tissues, which are mostly the core clock genes, and the rest of the circadian transcriptome, which covers about 2,000 genes in some organs, will be tissue specific and be important for the circadian physiology that is regulated by a specific organ. So what we see here is that we have our master clock and our peripheral clocks, and in both of them have a very large circadian transcriptome. So what are differences between this central clock and peripheral clocks? Because we've heard that the central clock is the master clock and actually is orchestrating all the others. So how is this working, and what are the differences between them? So in the central clock is located in the neurons of the SCN and is daily reset by light. And on the other hand, we have peripheral clocks, which are present in all other tissues and organs. And these are daily reset by stimuli, which are coming from the SCN. but they are also actually responsive to stimuli that come from metabolism, for example, hormones, which are induced upon eating, for example. If we now make use of a reporter animal, so if we have a mouse which contains a luciferase reporter gene, which is being transcribed under control of one of the clock gene promoters, you'll be able to follow luciferase oscillation in a certain organ. And what we see here is that ex vivo, an SCN slice will keep on ticking basically forever. And if you have ex vivo a slice of, for example, a liver, what you see is that this slice initially is oscillating, but after a few days, the rhythms start to dampen. And if you change medium, for example, you'll see that the rhythms will restart again until they dampen. So molecularly, this reporter seems to be working similarly in both tissues, but the SCN is capable of maintaining synchrony within each cells, and the peripheral clocks are not. So what happens in cell culture, for example, with liver cells or ex vivo with a liver slice, is that at the moment that you generate the cultures, you actually have cells which have all the molecular clocks synchronized as they come out of the body. But while in culture, and once they miss all the signals that come from the SCN, they will end up desynchronizing, and each cell will be at its own time. And what was observed already also more than 20 years ago is that if you change medium, you will be able to resynchronize these peripheral clocks. And what this means is that each cell is ticking at its own time, and at the moment that you give a stimulus, which is present in the serum in this case, of the medium, you will bring all the clocks to the same moment. So it's important to realize that there are several compounds like serum and forskolin, dexamethasone, which is a glucocorticoid, which are able to synchronize clock gene expression in peripheral clocks. The SCN is normally synchronized by light when in the animal, but that light signal is later transduced to glutamate, which is released, and this glutamate that's actually synchronizing the SCN clocks. And what's also very important to realize is that peripheral clocks and master clocks are composed of the same sets of genes and proteins. So the core clock present in the master clock and our peripheral clocks is actually the same. And so when we talk about a cell culture or a slice which is desynchronizing, what it means is that each cell starts now ticking at its own time, and by adding a compound we can resynchronize this. And the whole synchronization is not more than just a kind of signaling pathway. So I would like to depict the signaling pathways that are involved in this synchronization so that you understand what happens at the molecular level in terms of the core oscillator with synchronization. So what we see here is what happens when we synchronize the SCN, and in vivo this happens via the photic input via the retina and the retinal hypothalamic tract, which ends up in glutamate release in the SCN, which then will activate a signaling cascade, which finally leads to activation of gene expression. And one of the genes, which is important in this case for the molecular oscillator, is PER2. So basically, the light synchronization, which is translated into a glutamate release, which synchronizes the SCN, comes down to an increase in expression of PER2, which brings the molecular oscillator to a position where PER2 is high and can repress, together with cry, clock, and BMAL. And what's interesting is if we think of the other synchronization pathways which occur in the peripheral clocks, and I mentioned forskolin, which acts via the adenylate cyclase receptor, or dexamethasone, which is a glucocorticoid and will act via the glucocorticoid receptor. Both of these compounds will activate different signal transduction pathways, but in essence, it all comes down to activating transcription of the PER2 gene, both for forskolin and for dexamethasone, which is similar to what happens in the SCN when it responds to light input. So basically, a synchronization stimulus will lead to increase in PER transcription, which means that the inhibition loop can start again and all the cells will be at the same time. And what does this mean? If you want to study, for example, molecular clocks in culture and want to understand how they are synchronized, I would like to show you an example of SCN slices from luciferase reporter mouse, where the luciferase is driven by the PER2 promoter. We see here the example of 3 slices from 3 different mice, and they follow a certain oscillation. And now if we would like to see what the effect would be of giving glutamate to this cell, to these slices, so could we synchronize, or basically could we shift this oscillation by adding glutamate, what would happen? So I think here we have to go back maybe to the seminar of Joka Meyer. She has talked about the phase response curve and how you can shift the clocks in behavior by light. So I would like now to show how we can do that at the molecular level. So we see here again an example of an SCN oscillation from a reporter mouse. And basically there are different zones within an oscillation where your synchronizer, in this case glutamate, could or could not have an effect. So we know now that synchronization will occur by increasing the level of PER2. So basically, if you are at the moment in the oscillation where PER2 is already very high, you shouldn't expect an effect of synchronization. If you are at a position where PER2 is very low, you should expect a very high increase of PER2, which would lead to a change, in this case, an advance of the phase. And if you would be in an area where PER2 was high, but is actually going down, and if you suddenly increase it again, it gives the signal to the clock, something like the clock goes back again. And I'll try to go into this into more detail. So we see here again the same example, and now I depict an experiment which was done to try to see if we could catch our slice in a moment that it was unresponsive to glutamate. So basically glutamate was given here at a moment where PER2 was rising. And what we see is if we compare the distance between these 3 peaks and the same 3 peaks, but including the time of shift, we see that the difference is, the distance is more or less the same. So we have more or less the same period before and after, meaning there was no shift. If we now try to catch the oscillation with our glutamate pulse in an area where PER2 is already starting to go down, we see that we give glutamate at this point, and we get again, of course, a high rise in the PER2 levels. And what this means is that while the distance between these 2 peaks was 46.5 hours before the pulse, if we analyze the distance between peaks after the pulse, we have now a phase delay of about 1.5 hours if you count per peak. So you see that giving a pulse at the moment that PER2 is starting to decrease leads to a phase delay. And now if we try to give a pulse at a moment when PER2 is very low and should lead to a phase advance, we see that we actually can also do that. And that while the distance between these peaks was 46.6 hours approximately, it is now, when you give a pulse to advance the clock, only 43.3. So basically what I wanted to demonstrate here is that we can phase shift behavior by light by giving light pulses at different times of the day or the night. And the same type of effect can be achieved in vitro. And in this case, we are not looking at behavior, but we are looking at shifts in the molecular clock. So you can also basically create the same type of phase response curve, which Joka Meyer already showed before. But now, instead of looking at light effects, we are looking at chemical synchronizer effects. And another example, which is also very nice, is that also a few years ago, it was also found that not only chemicals such as forskolin or dexamethasone can shift the circadian oscillator, but also, for example, gamma radiation, X-rays, can also lead to a shift in the circadian oscillation. And by doing similar experiments, as I just showed with the SCN, it is possible to also create a phase response curve, in this case, for the gamma radiation, where you see that you can have moments where you have larger phase delays, advances than at other moments. So basically, what we observe in terms of behavior for the circadian clock at the macro level can all be also reproduced in the micro level if we are looking at the molecular oscillator. So now I would like to shift to seasonal clocks, and I think I should speed up a bit because we've heard also earlier that circadian clock is very, very important to encode season behavior. And I would like to briefly show that at the molecular level, the molecular clock is also actually responding to seasons, and that different areas of the SCN have different transcriptomes related to the seasons. So we see here a depiction of the SCN with rostral and the caudal part, which is anterior posterior. And if we look again at the reporter mouse, we see that depending on the photo period, if it's either summer with 18 hours of light, or winter with 6 hours of light, or spring or autumn with 12 hours of light, we see that this reporter, the PER2 reporter, will be oscillating differently in each of the photo periods. So the SCN is very small, but still anterior or posterior regions respond differently at the molecular level to the seasons. And not only the SCN is in this case split into rostral and caudal, but within the SCN you still have subregions where you have the core and the shell, and these are also having different responses in gene expression to the different seasons. And one study that was done to try to demonstrate the seasonal changes of the SCN was done using also a microarray approach, like I showed earlier on for the SCN and liver transcriptome. And here, basically, the idea was to expose animals to 3 photoperiods, winter, summer, and equinox, so basically autumn and spring. And by isolating SCNs using laser microdissection, would be possible to analyze the transcriptome of each of these parts of the SCN in different photoperiods. And basically what is clear is that there's in each, so rostral or caudal SCN, there's a large amount of oscillating genes, as we know. And we see that there are genes that have their peak time following the dusk time, so following lights off, basically. You have genes that will always follow midday in terms of peak, and you have genes that will follow lights on. And you can see that by the way they are shifting in their peaks while the animals are in different photoperiods. So this shows that also at the molecular level, we can identify seasonal responses of our master clock and our molecular clock to the seasons. And during aging, our circadian clock is also changing. And we have changes in behavior. Behavior becomes more fragmented, for example. What's also very interesting is that there are also changes at the molecular level. And this is just a very brief example. So basically, during aging, what happens is that many of the canonical circadian oscillator genes will lose amplitude. But what also happens is there's a very large portion of genes which normally does not oscillate or oscillates with a very low amplitude. And these will actually gain rhythmicity or gain amplitude with aging. This is an example from an aging study in Drosophila, but similar effects are observed also in humans and mice and other animals, showing that during aging, for example, what Jokemeyer showed earlier is that the neurons of the SCN lose synchrony, and that leads to a lower amplitude of oscillation of the SCN in general and of our behavior. But what happens at the molecular level is that a set of genes will lose amplitude and others will gain, which usually is related to, for example, response to stress and to other alterations that you happen with age. And I think I would like to leave this here. I had still a little story I wanted to tell, but I think I will leave that to a next time. So I would be happy to take questions.
Thank you very much, Inês, for this nice talk.